Compartments help specific reactions happen and minimize competing interactions. So when we look at our compartmentalized cell, we understand that the semi-permeable lipid bilayer acts as a barrier. Most of the time, we're thinking about these phospholipids only in the outer cell membrane, but the thing about eukaryotes is that there are membranes acting as barriers within each compartment of the cell. So we can look at specifically lysosomes as an example here. The things that happen within lysosomes should only happen in lysosomes. So the enzymes, these hydrolytic enzymes inside lysosomes, would be very dangerous to have wandering around throughout the entire cell. But these specific enzymes only work at very low pHs, so you have to have one isolated area with a really low pH and not the entire cell at a really low pH, because that would also inhibit many other reactions from happening. We can also look at mitochondria as an example here. There are very specific reactions that happen within the matrix or the inside area region of the mitochondria. So we have the initial splitting of glucose to pyruvate, but then all of the chemical reactions that happen in the citric acid cycle and the conversion of pyruvate to acetyl-CoA, those reactions don't have to compete with other reactions and other chemicals present if they are isolated within the matrix of the mitochondria. Also, this extra membrane is folded, which helps maximize surface area. If we understand electron transport chains and how we have protons getting pumped and feeding back through ATP synthase, if we have more folds, we have more pumps. If we have more folds, we have more ATP synthase, and it would be more efficient at creating ATP. So when we have all of these specific regions, they have specific jobs, and changes to those regions would hurt cellular function. If you mess up the structure, it's not going to overall work as well. If you don't have as many folds, if you have the wrong chemicals in the wrong places, if the enzymes aren't activated correctly, it's not going to work as well. So we compartmentalize these things, and they have very specific environments in which they're supposed to carry out their functions. So any changes to that would hurt cellular function. So again, we have compartments where specific reactions are meant to occur, and it minimizes other competing interactions by making it have a specific location where that can occur.